As always, thank you for being here despite the early time of day and the inclement weather. Much appreciated. Hello, those in Evans. Good morning, Anna. In you and I, as we were developing from a fertilized egg into you and I, genes were going on and off. And that, of course, is why we're not a clump of completely identical cells, but contain approximately 230 different cell types. And last time, I told you in considerable detail about the mechanics of gene expression. I told you about transcription that doesn't begin randomly but starts at a specific place in the genome, that uses one of the two strands of DNA to produce an unstable intermediate in the gene expression process, a messenger RNA. I told you that that messenger RNA made by RNA polymerase is then exported into the cytoplasm after it was acted on by a machine called the spliceosome, which cuts out most of it and puts together the bits that are ultimately translated. And translation is, of course, the act of transitioning from the language of nucleic acid to the language of protein. The messenger RNA is exported into the cytoplasm, is engaged by this marvelous machine called the ribosome, and the ribosome makes the protein. Today, I will tell you how that's regulated. But before we dive into this, I need to take the first couple of minutes of today's lecture to walk you through something I ran out of time last time for. Oh man, the hanging prepositions in English are something else. What happens when the DNA goes wrong? Wrongity, wrong, wrong. When there are changes in its sequence, what we get is we get a mutation. And I've told you before, and it's a very simple and useful number to remember. A large number of complete human genomes have by now been sequenced, and we know what the mutation rate is. There are about 6,000 coding changes between two randomly chosen human beings. So 33,000 genes, give or take about 10,000. But the number of coding changes, so changes that actually have an effect, 6,000. What kinds of changes? Well, I'm going to walk you through specific representative examples. And you know me, I love telling you what will be on the midterm. The mutation types and their names will be on the midterm, and I'm expecting you to memorize them. So a question on the midterm will be, here's a DNA sequence, here's a mutation in it, here is the way that it affects the protein. What is this type of mutation known as? So what am I expecting you to memorize? Well, here's a stretch of DNA, Watson, Crick. Here's a stretch of RNA. Here's the protein, the amino acid sequence that stretch of RNA encodes. The first one is the easiest, a silent one. We start with it. Where is this cognate? GGC. So we start with GGT, and then we get a single base change. This TA base change gets changed to a CG. The genetic code is degenerate. So different codons mean the same thing in specific cases. In this case, this codon, I suppose, means glycine, whether it's GGT or GGC. The change is there, but what the protein is does not change. That type of mutation is called a silent mutation. Are they completely inert, these silent mutations? Oh, far from. And if I have time at the end of this section, I will talk to you a little bit about the remarkably fun things that these supposedly silent mutations do, and how therefore challenging it is to look at the differences between the genomes of two human beings and say, the following 4 silent mutations are in fact quite loud. First type, silent. The DNA sequence changes, the RNA sequence changes, the protein sequence does not. What kind of event can lead to that? A change in the 3rd position of a codon in those situations when the original codon and its mutant allelic form encode the same amino acid. That, unfortunately for you and I, and for 80,000 of our compatriots who have sickle cell anemia, is not always the mutations that take place. Sometimes change in DNA sequence actually change the amino acid sequence that is produced after the mRNA is translated. Now, let me be very clear. The sequence that you have before you is not the sequence of alcohol dehydrogenase, nor is it the sequence of hemoglobin beta. It is just a representative sequence, transcribed and then translated. Why is there this title over this picture? Because if you were to look at the sequence of alcohol dehydrogenase in somebody from Northern Europe, thoroughly enjoying his mid-afternoon beer number 4, and somebody from southeastern China who turns red in the face from just a sip of that beer, you will see a similar type of change, a missense change, in which we go from, let's see, I should have aligned the wild type sequence first. So GGT CCA, and here it goes GGT TCA. So that base gets mutated to a CCA. That causes a substitution in the amino acid sequence of the protein. Instead of saying glycine, when I say proline, as you hopefully know by now, proline is a very different biochemical creature than glycine. The protein sequence is changed, and in many cases, its function is changed, as in the ultra-fast allele of alcohol dehydrogenase, as in the allele of beta-globin that causes sickle cell anemia. This type of change is called a missense. I'm expecting you to memorize it. Sir? The gentleman is asking a fantastic question. If the amino acid sequence changes, but the protein structure and function do not, do we still call that a missense mutation, or is that a silent mutation? For technical reasons, thank you for asking, that's still called a missense mutation. One of the additional challenges in human genomics is how to parse missense mutations that are actually silent, they don't really do anything, from those that do something. And we'll talk about that on Wednesday next week. Thank you for asking. Good for you. Missense mutations can cause new things to start happening. So the southeastern Chinese allele of alcohol dehydrogenase is faster than the northern European one. It can also cause disease. The sickle cell anemia allele of beta-globin causes sickle cell anemia in homozygous form. And in fact, there's a very nice picture in your book which distills the statement I just made to precise molecular detail. Wild-type beta-globin, the allele that causes sickle. GAG, as per the genetic code rules, encodes a glutamic acid. The allele that causes sickle is a single base substitution and causes very severe disease. Next, a mutation that causes the premature termination of translation. So instead of producing a full-length protein, Mother Nature simply stops soon after it sees that mutation. A truncated protein product is produced, and most of the time, in fact, its function is completely ablated. By way of an example, here is TCA-TGA. The wild-type allele is TCA-TGC. So that base pair is changed now to a AT. That, in genetic language, is a stop. Mother Nature doesn't know it should be producing the full-length protein. It basically does what the instructions say. It attempts to translate this. It sees a stop at that position, and it stops. An example of such a nonsense mutation is an allele of BRCA1 that causes heightened susceptibility to breast and ovarian cancer. Thus, we have silent, no effect on amino acid, missense, a different amino acid, nonsense, ablation of translation. Translation can be ablated in one other way. We can shift the reading frame. Please write this down. Shift the reading frame. Put a question mark. and then a little nota bene. Homework to look up what that means. Please. It's very straightforward, very well explained in your book. Ask yourself, compare and contrast nonsense and frame shift mutations. The outcome is the same. The path to the outcome is different. Et voila. There's a survey of the types of mutations that you and I experience, and that I'm hopeful you will be able to basically regurgitate next Friday or whenever the midterm is. And now, woohoo, my favorite bit. Finally, the regulation of gene expression. I promised you last time I talk about this, but then I spent the entire lecture talking about the biochemistry. I wonder what that is. Static off of me? Static in the air? Be afraid. Be very afraid. Wow. Okay. All right, let's try this some more, and if this keeps happening, we'll try to fix it some other way. Can you still hear me in the back? All right. Once upon a time, you were this. If you go like this, what is happening is this is firing. This is a seriously, be nice. There are like 450 students here. They came here early. This cell in front of you is firing, and it helps you coordinate that motion. It's a Purkinje neuron in your cerebellum. The reason it looks so dramatically different from this is it expresses a different set of genes than this. Now, I've told you about the mechanics of gene expression. I told you about transcription and translation. My point to you this morning is that this image shows what happens, but says absolutely nothing about what controls what happens. So in an analogy that I will make throughout this morning's lecture, think about the general elections that have transpired in your lifetime. The United States of America, its population, went to the polls, and in 2000 and 2004 elected George W. Bush, and in 2008 elected Barack Obama to be the President of the United States. Now that describes what happened. The question is why? What were the mechanics? What was the regulatory process that caused the people of this country to vote for one individual in one election and for a different individual in another? Well, this schematic says absolutely nothing about that. Similarly, in studying genome regulation, the number one challenge at this point is not to describe what happens, but why that happened. And I'm going to try to walk you through some of our current understanding of that and conclude with how little we actually in fact understand about what's going on, in the hope of inspiring you to pursue that as your independent career. I rewrote figure 16-6 because I just rewrote it. I want you this is the version of it that I need you to know. What does the word know mean? Know means simply understand what it says. Gene expression is a multi-step process. It starts in the nucleus, continues after transcription and splicing with the export of the messenger RNA into the cytoplasm, which is then translated, the protein is made and goes on to do its thing. At which levels in the process is there control? So as one thinks about the Purkinje neuron, at which levels in this sequence does it control its gene expression? At all of them. An enormous amount of effort Mother Nature puts into determining which genes are transcribed and at what rate by regulating initiation, elongation, and termination of transcription itself. The rate at which pre-messenger RNA is made into messenger RNA and how stable it is exquisitely controlled by regulating pre-messenger RNA's processing, messenger RNA export, and stability. What does control mean in this context? It means it differs from cell to cell. Why? Because Mother Nature has a mechanism to have it differ. She spends a lot of time focusing on the rate at which proteins are made. So will a messenger RNA be translated, and at what rate? Finally, putting it mildly, last but not least, Will the completed protein be available to do something? Whether the protein folds correctly, whether it is directed correctly to where it needs to work, and how stable the protein will be, is under exquisite regulation from the cell. These multiple levels of control marvelously all integrate into producing the Purkinje neuron, or the muscle cell. Now, this word is underlined. By the way, what would I like you to know about this for the midterm? You know, I'm more or less expecting you to understand all of this. So, do I need you to memorize this? No, it would be kind of unfair. The midterm is in a week. But I do need you to read this slide pretty carefully. Messenger RNA was actually discovered in the 1950s. And the reason the word transcription initiation was underlined in the previous slide is for the following reason. Are there what seems to be like 20 different types of gene expression control there? However, most work has been done on, and most is known about, the regulation of gene expression at the level of, mental drum roll, regulation of initiation of transcription by RNA polymerase. This doesn't mean that the other steps are not important. They are. It's just that we know the most about that. And in fact, I will spend the rest of the lecture talking about nothing but that. Now, only about 10 years ago, or maybe even 5 years ago, this would have been unacceptably biased. I mean, how dare you? We understand you got your PhD on this and have been working on this for 2 decades. Why are you indoctrinating the helpless students with what you like? Well, before we dive into this, let's take a quick quiz, and then I'll answer the question. All right. As always, I promised you a question on this. Spot the error, please. A, the promoter is a genetic element that specifies the position along the DNA where RNA polymerase is to bind the DNA, unwind it, and begin transcription. B, in contrast to DNA polymerase, RNA polymerase doesn't require a primer to initiate synthesis. The first nucleotide then is just put down. RNA polymerase, just like DNA polymerase, says option C, synthesizes a nucleic acid in the 5' to 3' direction. The distinction being, of course, RNA polymerase uses ribonucleotide triphosphates, not deoxyribonucleotide triphosphates. As RNA polymerase elongates along a gene, says option D, the bubble of unwound DNA in its active site contains an RNA-DNA hybrid helix. And finally, because of Watson-Crick base pairing rules, the sequence of the RNA transcript produced at the conclusion of transcription is the sequence of the DNA template strand, other than the replacement of all T residues with U residues, of course. Okay. Start session. All right, let me put the question back up so you can see it. And with that, folks, let's take maybe 2 minutes to do this, maybe a minute and a half. You know the rules? It's like Calvin Ball. There are no rules. All right, we're halfway there. Thirty seconds. All right. 5, 4, 3, 2, 1.5, 1, 0.5, 0.4, 0.3, okay, 0. All right. Let's see if it was worth your time to come through the rain. Great job. Good for you. This was actually reasonably subtle. Good for you. I'm impressed and happy. Thank you for noticing. Thank you for listening last time. Thank you for studying. Good job. All right. Go away. You've done your job. Now that we know that you know the mechanics of transcription, I need to show you an example of why this was worth it. I need to show you Shinue Yamanaka's unbelievably quixotic experiment. And yet it succeeded. It revolutionized modern biology. I would have bet, I don't know, 1,000 rubles against this experiment working. Now, don't ask me what the current ruble to dollar exchange rate is, but I would have still bet 1,000 rubles. Shinue Yamanaka showed that you can take a differentiated human cell, specifically a fibroblast, a mature cell with a specific phenotype expressing its very specific genes, and convert it into a stem cell. What is a stem cell? A stem cell has three properties that make it markedly different from a fibroblast. First of all, it is immortal. It can divide forever. A human fibroblast, if we harvest me, homogenize me, and put me, the homogenate of me, thank you for not going, oh, homogenize him. You put me in culture, all of the cells will die. The only cells that will actually grow out, more or less, will be the fibroblasts. But they will only divide about 30 to 40 to 50 times. And they will just senesce. They'll stop. Stem cells divide forever. Second, not only do they divide forever, they forever maintain their identity through that division. So they're eternally self-renewing. Last but not least, stem cells, at least the stem cells that Professor Yamanaka made, are pluripotent, so they can differentiate into all the cell types of the adult body. So they have endless potential written into them. How in heaven's name would you convert a fibroblast, a differentiated cell, into a stem cell? By rewiring its gene expression program. How would you do that? By expressing in it, so making a lot of, in that fibroblast, 4 proteins. What are those 4 proteins? They are transcription factors. What's a transcription factor? A transcription factor is a protein that binds DNA and changes gene expression. This remarkable experiment demonstrated, and I don't have any time to walk you through the data, but I encourage you to pull up this astonishing paper and read it. It basically shows that transcription factors are dominant with respect to cell phenotype. Not just dominant, that you can take a fibroblast and convert it into a stem cell by rewriting its gene expression program. The DNA is the same, the phenotype is different. Why? The gene expression program has changed. This is why there is so much focus on the regulation of transcription at the level of initiation by RNA polymerase, because that's what many transcription factors do. That's the process many of them control. I want to point out that your textbook contains, and I'd like you to please look at it, an early important example in which a transcription factor was used to take cell type A and make it into cell type B. It describes a transcription factor called MyoD. The textbook explains how during early development of you and I, that transcription factor, that protein, is made, goes into the nucleus, changes gene expression, and muscle cells are formed. What your textbook, I don't think, mentions is the result of this remarkable paper from the, I believe, mid-1980s, which showed that if you take that transcription factor, which makes muscle cells in normal development, you can convert poor fibroblasts to muscle cells as well. Taken together, these two pieces of data argue very strongly that my focus and that of hundreds of thousands of scientists around the world on how transcription factors do their thing is amply justified. So, transcription factors are kind of like the voters in a general election in a normal democracy. Of course, there are huge numbers of factors that contribute to its outcome. But at the end of the day, whatever the voters say goes. Hence the focus on them. I need to show you a few biological structures because they're actually quite remarkable. Figure 69 shows you three characteristic structures of transcription factors: a helix-turn helix, a zinc finger, and a leucine zipper. The only phrase that you need to know is the phrase zinc finger. It is the most abundant type of transcription factor in you and I and in everything else. And I like this picture because it simply shows that there you go. There's a protein bound to DNA. But it's not just stuck to DNA. It is bound to a specific sequence. It recognizes a specific statement in your genome. And it is that sequence-specific recognition that explains why transcription factors are so powerful. This allows me to remind you of the structure of DNA and then show you these structures in more detail. Before I do that, I cannot not acknowledge the founding fathers who have made the world that we live in, conceptually speaking, possible. The zinc finger was discovered by Sir Aaron Klug. Its structure that I'm about to show you was solved by Carl Pabo. However, the first human zinc finger transcription factor was purified here at Berkeley in Robert Tijan's lab. So we have many things to be proud of, and this achievement is one of them. Without further ado, let me show you, first, remind you of what this statement means. Zinc finger proteins and many other transcription factors bind DNA in the major groove. Excuse me, where is the major groove? DNA. I'm going to rotate it sideways, and I've done this for you once before. There is the large indentation that runs along its side, and there is a smaller indentation that runs along its side. One more time. One groove runs up here, one groove runs on the other side. There is the major groove, and there is the minor groove. Now I can show you how a zinc finger binds DNA. I know. Isn't that amazing? I agree. Can you see which groove it is bound in? Major. Yeah. Let me spin it for you so you can. Let's do. This is worth it. This is the most important type of transcription factor in you and I. Movie, program, camera, loop. Y roll. Y roll. How about 8 seconds? Go ahead. This is what powers you and I. The fact that this remarkable protein can recognize a specific stretch of DNA by binding to it in its major groove. Why major? Because it's the bigger one. There's more room to fit. How do proteins bind specific sequences in DNA? That's a perfect place to pause. There is an alpha helix, which you've learned about from Professor Malkin. And the alpha helix has residues projecting from it, amino acid side chains. And those amino acid side chains make hydrogen bond, electrostatic, and van der Waals contacts with the basis of DNA. So it's just like Braille. When a non-sighted person runs his or her hands down Braille text, he or she feels the bumps and the indentations. That's how zinc fingers bind DNA. So the simile is quite apt. Let me show you how that works. I'm just going to zoom in on an alpha helix, recognizing a specific sequence of DNA. Whoosh. There is the amino acid sequence RSDELTR, recognizing the DNA sequence GCG by going. And there is a different amino acid sequence recognizing a different DNA sequence. So literally, the protein goes like this. What are you? GCG. Good. You're my gene. Will then any of this be on the midterm? Don't panic. It won't. The word zinc finger will be. Will the structure be? No. I need to walk you through the biological problem, the solution to which I'm about to describe to you, which is that in the genomes of all living forms studied, there are genes whose activity changes as a result of the change of a titer of some molecule provided by the environment. This will not be on the midterm, but will be on life. I do need to show this to you. Those of you who haven't seen this, prepare to gasp. But I suspect that many of you have. These are obesity statistics for the United States. Blue is, let me pause this for a sec. So blue is 10 to 14% obese individuals. Dark blue is 15 to 19%. This color, I'm partly colorblind, I don't know what color that is 20 to 24%, and this color is more than 30%. 1989, this is what the United States looked like. 90, 91, 92, 3, 4, 5, 6. Okay, we have our first states with more 1 and 5. Yep, 1 and 4. I think Colorado will be the last holdout. Colorado shall fall. From a standpoint of these data, I mean. This happened in our lifetime, folks, in the United States of America. Why am I telling you this? Because there's nothing I can do to change the way people eat. But I can tell you that the extraordinary prevalence of both type 1 and type 2 diabetes in the United States as a result of the fact that we went from this, from what we just saw, to this, is due to small molecules changing gene expression. And this means we can counteract the occurrence of type 1 and type 2 diabetes by using different small molecules. And that in fact, is a major direction of drug development in the United States today. And some of you, I'm hopeful, will become part of it. I was in DC once, and I took a picture of this. This is an ad for a sandwich shop called Potbelly. They advertise sandwiches called New Bigs, same deliciousness, now 30% bigger. I mean, I agree. So this is both A, funny, B, tragic. I mean, this is in a country that went from this to this in 18 years. 30% bigger. I showed you this picture. This is in this process. Anybody remember what is the small molecule that causes this process? I'm glad. I actually didn't mention estrogen. Progesterone is what I said, but estrogen causes this process as well. What I didn't tell you is estrogen is carcinogenic for the breast. So not only is estrogen morphogenic, so it causes this, high doses of estrogen, are not high, just doses of estrogen are known to be carcinogenic for the breast. This creates a general picture of the way Mother Nature has chosen to do her thing. Whether a tadpole metamorphosing into a frog, a pupa of an insect metamorphosing into a fly, or breast epithelium responding to estrogen by converting to breast cancer. This is all changes in a gene expression program driven by a small molecule. On Monday, I am incredibly excited to report that I was able to arrange for an extraordinary guest lecture. You will hear about the next generation of approaches to cancer from Dr. Sophia Mond from the Stanford University School of Medicine. Please be on time. Don't forget your clickers, but please be on time. I promise you, you will never forget her talk. I saw some of the data, and I'm still trying to find my lower jaw. Why am I telling you this in the context of this? Oh, this is like, wow. Why am I telling you this in the context of the schematic I just showed you? Because she will describe to you how hormones cause cancer and what we can do about this. Briefly, furthermore, you will hear a lot more about this from Professor Firestone in the third section of the class. So this guest lecture is talking about this for a pedagogical reason as well. I now have the great joy of spending 10 minutes walking you through something that many of you, I think, have already learned, which is the marvelous, beautiful, amazing work from François Jacob and Jacques Monod that provided the intellectual foundation of how we think about how small molecules change gene expression. In brief, I think this is taught in US high schools. Is it? It's not? Oh, you're lying. You just want me to go slower. Okay, I beg your pardon. I retract the you're lying statement. I am misinformed. Well, what they basically showed is that genes can be under positive regulation and negative regulation. Specifically, that there are proteins called activators that turn genes on, and that there are proteins called repressors that turn genes off. And that the way that the genes respond to hormones, small molecules, actually works, the DNA itself doesn't respond. RNA polymerase doesn't respond. It's these regulators that respond to small molecules. That's the mechanism. This was, my goodness gracious me, this was 50 years ago, half a century ago, and still is remarkably influential. Now, the model system that they used to study this is how the bacterium E. coli that lives in your gut, Escherichia coli, breaks down lactose. You've actually already seen this schematic. I talked to you about lactase persistence for a very good reason. I wanted you to learn about lactose early. This enzyme in bacteria, beta-galactosidase is its name. Why is it not called lactase? To make your life more difficult. It's a different enzyme. Does it perform the same chemical reaction? Yes. Beta-galactosidase breaks down in the bacterium the sugar lactose, and galactose and glucose can then be burnt. The addition of lactose to the medium induces the synthesis by the bacterium of the enzyme to metabolize that sugar. Why? It will be wasteful to synthesize an enzyme in the absence of its substrate. This is a resource conservation adaptation. Note, humans are different. I told you that some adults are lactose intolerant and some adults are lactose persistent. If that's your genotype and phenotype, as highlighted here by Alex Morgan, go Bears, and Jeremy Lin, then that's your phenotype for life. No amount of drinking milk in a person who doesn't have the lactase persistent phenotype will induce lactase synthesis. It will induce a number of other things, but not lactase synthesis. So in that regard, bacteria are different. They are able to turn the gene on and off on demand, depending on the composition of the medium. Voilà, mesdames et messieurs, the Jacob-Monod model of the lac operon and its control. Let me walk you through it. Do me a favor. Put down your pens. Don't write anything down. This is beautifully explained in your book. But I want to explain this to you once properly so that you pay attention in real time. The E. coli genome encodes 3 genes, the products of which are required to break down the sugar. This one, and that one. They're called LAC-Z, LAC-Y, and LAC-A. Together, they are necessary for the bacterium to eat this sugar. Notice that they are together. They're together in the genome. In fact, they are translated from the same messenger RNA. This is a beautiful adaptation to make sure that they are under coordinate control. The bacterium either needs all 3 of them or none at all. That's why they're translated from the same messenger RNA. Humans do this near never, with very few exceptions. A given messenger RNA produces only one category of protein, one type of protein. Now, there could be different forms, but it'll still be the same protein. These polycistronic messages are a hallmark of bacteria. How is expression, how is transcription of this gene directed by lactose? Well, this is where the gene starts, and this is where the gene ends. And this is where the regulatory circuitry comes into play. Specifically, in addition to the gene itself, to its coding sequence, the bacterial genome contains sequences that control gene expression, or regulatory sequences. And the 2 types that I need you to know are the promoter. You've already learned about that last time. This is where polymerase shows up and says, unwind, engage. And something called the operator. The operator is a really fascinating piece of DNA because while the promoter, of course, as you know, binds RNA polymerase, the operator binds a regulatory gene. So it binds a transcription factor. I just showed you how in the human genome, a zinc finger protein binds DNA. In the bacterial genome, a different kind of protein, also binds DNA in the same manner. Structurally, it's a different type of protein. But the fundamental idea, protein binds to specific sequence in DNA, is exactly the same. Well, why would it bind there? If it were to bind there, polymerase would be incapable of transcribing this gene. Let me play out the complete sequence for you. Under normal circumstances, in the absence of lactose, the inducer, the sugar in the medium, the gene is off. None of these three proteins are produced. Why is that? Because the lac repressor protein produced by a different gene in the bacterial genome actually binds to the operator and blocks transcription. How does the bacterium induce expression of the gene upon addition of milk to the medium? What happens? You take some E. coli, put them in some milk, they go, ha, 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 got milk. What happens next? What happens is this. I decided to not animate and just show you one important difference. When you go home and study this over the weekend, basically look at this and ask yourself what changed. What changed is the appearance of the sugar. The sugar we have now renamed and are going to call it the inducer. Now, there's actually an additional chemical step here. It doesn't matter. From your perspective, what happens is the agent in the medium, the sugar, that should cause this gene to be transcribed, binds to the repressor. And that binding inactivates it. So it shuts down the repressor. The repressor falls off the DNA, and RNA polymerase goes, ha, ha, ha. A simple two-step sequence. Now, to you, this will be like, that's it. In bacteria, it's that simple. And I say in bacteria because, as I'm about to show you, in you and I, it's anything but. Do we do this? Yes. But first, I need to tell you that what I would like you to please do is, now that you've listened to me explain how the lac repressor negatively controls transcription of beta-galactosidase and the two other genes, I'd like you to study on your own time how a protein called CAP positively regulates transcription of this gene. I need you to please study this on your own time. It's very simple. The logic is very related. But what I do want to tell you now is why this machinery exists. The logic is glucose is a better energy source than lactose. In other words, if you take E. coli and you put it both in milk sugar and glucose, the bacterium doesn't need to metabolize lactose. Remember, it's a microbe. Its goal is to divide as fast as it can and take over. How does that work? There is a system, in addition to a system that senses lactose, the milk sugar, there is a system that senses glucose. And that sensor also works via a transcription factor. So the beta-galactosidase gene is controlled by not one, but two separate transcription factors. One determines whether or not there's lactose in the medium, and one determines whether there's glucose in the medium. And they talk to each other by controlling whether or not RNA polymerase can transcribe. And that's what I'd like you to know for the midterm. Does this happen in you and I? Yes, but by analogy with elections. There is elections at Berkeley. There are elections, right? The ASOC has elections, and there's Vishali Lumba. She's our president right now. But there's also the US general election, which you go from that, via that, to the president. Why am I telling you this? Much like the elections in the United States happen at much larger scale, there are processes, inputs, and phenomena that simply don't occur at the scale of a public university on the West Coast. What happens in you and I with respect to gene regulation involves elements of what happens in bacteria, but embellished to a point where it's no longer clear whether we are to keep thinking about gene regulation in you and I using the simple concepts developed in bacteria. That said, we have to start somewhere. So what can we start with? Well, by the way, starting from this, the only things that will be on the midterm will be the figures in the textbook. I will rant briefly in another 2 minutes. That won't be on the midterm. What would I like you to know? The following facts. Human genes don't just have promoters. They have additional regulatory elements, specifically these things called enhancers. I taught you about lactase for a reason. Remember where the mutation that encodes for lactase persistence, Alex Morgan versus Jeremy Lin? It occurs in the enhancer of the gene. Second, now, of course, the core promoter is also bound by transcription factors. There are so many more of them. It's slightly dizzying. First, this is something I need you to know. The core promoter is bound by a transcription factor called TBP, Tata, box binding protein. And an army of additional transcription factors. So a lot more, and I'm about to show you how many more. And the enhancer is also bound by transcription factors. And then there are bridging entities that help the enhancer talk to the promoter. Furthermore, some genes are bound by, let's say, transcription factors 2, 5, 7, and 8. Those are not really their real names. A different gene in that same cell could be bound by transcription factors 1, 5, 8, and 11, producing gene expression. Thus, human genes have multiple regulatory elements. They're bound by multiple transcription factors that somehow come together to work. That's what I need you to memorize, because this will be in upper division classes, etc. And now the inconvenient truth. Bear with me for 2 minutes. This will serve you into grad school and medical school. This is pure Calvin Dadism. Calvin asks, Dad, how do they know the load limit on bridges? And Dad says, oh, they drive bigger and bigger trucks over the bridge until it breaks. And then they weigh the last truck and rebuild the bridge. And Calvin goes, I should have guessed. Mom goes, dear, if you don't know the answer, just tell him. The schematics that you see here, in particular this, is pure Calvin Dadism. We don't actually know what exactly happens. In particular, what we don't know is how it all comes together. OCT4, the one human gene I need you to know, is the master regulator of human stemness. Here's our current model of how it is regulated. Nobody likes this model, but this is our extent of knowledge, this sort of car pileup or pile of children's toys sitting at its promoter. Our current model is a little bit like this picture of the UC Symphony, which just played Shostakovich 11, I should point out. There is a bunch of people on stage, but we don't know who plays which instruments, and that's how each individual contributes to the overall sound being produced. Just like in elections, the question becomes, how are the multiple inputs in this remarkable country combined to produce a specific president? The challenge for your generation is to understand how the following things are integrated. I have 60 more seconds, and then I'm going to let you go. Chromatin. Jasper Ryan and Robert Tijan are the world leaders in the study of how chromatin and gene regulation come together. There is no good model. There will be by the time some of you are professors or physicians or public health professionals. Small RNAs. Lin He at Berkeley is a world leader in understanding this, which will be clear by the time you graduate. A wonderful model system to start your study of this, transcription regulation and development. Mike Levine and Mike Eisen, #1 and #1 respectively in the world study of this problem. Please be on time for Monday's lecture. It will be on the midterm. There will be a quiz, and I have office hours now in 18010.